All right, I guess we'll get started. So it's my pleasure to introduce today's speaker, Quavi. He's an assistant professor in residence in the Department of Pathology and Laboratory Medicine here at UCI at the Medical Center. So he has joint appointments also in chemistry, an active member of several of the research centers here on campus, including the Cancer Research Institute, the Institute for Immunology. He's a MD PhD. He has his MD from University of Illinois at the Carl. Do you get it at the Carl?
So it was
UIC.
It was UIC,
UIC,
but
okay.
yeah, still work closely with Carl.
Okay, got it. So he's up in Chicago for his medical degree, and he got his PhD from the University of Illinois, Urbana-Champaign. And before that, he's actually here as an anteater. He got a dual degree in chemistry and biochemistry here. So proud anteater. That. And so his research is an interesting combination. So it involves microring resonators, also looking at virology, optical biosensing, translational diagnostics. And so really looking forward to just kind of this, as a type of research using optics that we don't normally think about in terms of looking more at biodiagnostics. And so with that, let's welcome Abe.
Thank you so much for the introduction and the opportunity to present today. So what I'm going to be discussing today is really how we're leveraging optics and different techniques on controlling and manipulating light directly towards clinical diagnostics. So I am a practicing physician. It pays the bills. 1 day a week, I'm at Orange signing out cases. And a lot of what you're going to see today is stuff that we're trying to integrate directly into our clinical pipelines using this work. Just really quickly, disclosures. I do have some patents related to one of the companies you're going to see today, Genelite. My car is 20 years old. You can make your assumptions of how much money we've made off of, the patents with them, but just needed to throw that out there. And then learning objectives asked in case we needed CME, but we don't necessarily need that. The broad theme of what I'm going to show today is when we can combine unique advances in optical technologies, combine laboratory medicine, which is my medical specialty, and unique biomarkers, we can start building facile detection pipelines. I use the term pathogen X a lot. And the idea with pathogen X is that what you're seeing today, we're going to focus on a few niche systems, but we can apply these towards a number of different diseases and a number of different targets relatively quickly, which is ideally what we want, as we've seen with this last pandemic. To take a step back, we need to just briefly talk about current detection paradigms in the analytical space. So if you walk into our clinical operations at Orange right now, these three tests are some of our bread and butter. We've got PCR-based techniques, you've got ELISAs, and you've got lateral flow assays. And while these are our bread and butter techniques, they have disadvantages that include limited multiplexing capabilities. In many of these, especially lateral flow assays, you've got limited quantitative information. So you get a yes or no answer, but you can't really trend data or patient prognosis. You've got centralized laboratory needs. So we can run these in a centralized lab if you send it to Orange, but you can't actually run these in the field. Or if you have the issue of cold chain custody. And what we take for granted in terms of refrigeration and electricity, if you take this out to the field, are going to be huge issues. So these are things that we're really trying to address in these gaps. The first part of the talk, I'm going to talk about how we're going to leverage photonics and unique biomarkers to address a really critical gap in the diagnosis of filoviruses. And filoviruses, the most famous being Ebola virus. I'm just going to refer to it as Ebola. The formal name now is orthoebolavirus ziarescens. But when I say Ebola, that's what we mean. We need to take a step back and actually understand how we diagnose it right now to figure out what the gaps are and what we can actually target. And so what I've highlighted here is the initial presentation of Ebola virus. You start off with an infection. You then have an incubation period from 2 days to 3 weeks. And in that incubation period, you either have no symptoms or you're very nonspecific symptoms. You then have the symptomatic period, which can last up to 2 weeks. And finally, you have resolution if you do survive. Now, what I want to highlight here is if you look at the symptoms that you commonly get, you have increased body temperature, so fever, fatigue, nausea, vomiting, diarrhea, headache. These are all very nonspecific. You don't know if you have Ebola virus or if you have the common cold or you have malaria. When you think Ebola, you think of bleeding. That's only in about 20% of the cases. So on symptoms alone, it's a terrible disease to diagnose. This is a poster from the 2015 outbreak, the largest outbreak to date in the DRC by UNICEF. And it once again highlights these very nonspecific symptoms you see following infection with Ebola virus. So once again, on symptoms alone, terrible disease to try to diagnose. What I've highlighted here is the different diagnostic tests that we have as a function of infection course. On the x-axis is days post-infection, starting minus 21 days. 0 is when you start seeing symptoms on the x-axis. And you can see that there are several techniques. We've got RT-PCR, antigen assays, serological assays that all come at different periods of time and work at different periods. What we're really interested in and what we really want to target is this phase where you have either no symptoms or you're very nonspecific. And we call that as the window period. That's the period in which we don't have any diagnostic or you don't have symptoms that can really give you a definitive diagnosis. And that's really what we want to be targeting when we're reinventing these clinical assays. So we get asked this every day at the hospital. Why does it matter? Why do we care? Number one, it's earlier initiation of supportive measures, and we'll show you that just earlier diagnosis leads to better outcomes. As we saw even in the US with the pandemic, you have allocation of healthcare resources. So you need to know who's infected to see who gets the treatments or who needs to get admitted to a unit. And finally, you can do contact tracing and isolation. So prevent a pandemic or an outbreak from becoming a full-blown pandemic. How do we actually currently manage Ebola virus? Mortality is pretty terrible. It's up to 90% depending on the strain. And really, current paradigms for treatment are supportive measures. We give you IV fluids, we treat your symptoms, and we try to treat any comorbidities you might have. That is standard of care right now in the world. We do have a number of biologic-based therapies, and in fact, we work with MAT Biopharmaceuticals on some of the work you'll see. But in general, you just need to give IV fluids and pain management, and that's our current paradigm. Shown to the right are two curves. In orange are patients who were in an Ebola treatment unit. So we said we assume you have Ebola. We're going to give you IV fluids. We're going to treat you aggressively. In blue are patients who were in a non-Ebola treatment unit, so a general hospital in the field. And the y-axis is your survival probability. So pretty much just by going into a much more aggressive treatment course, your survival's improved just by earlier initiation of IV fluids. So clear benefit in terms of just getting this earlier diagnosis and treatment. To understand the biomarkers we're going to use, we need to understand the structure of the Ebola virus itself. And Ebola virus is only 7 genes. In particular, highlighted here in green is the gene for the glycoprotein, the structural protein that's going to form the envelope and actual viral structure. Now, about 30% of the time, there's a transcriptional stutter in there that's removed, and you have the full-length glycoprotein that's generated. However, 70% of the time, that transcriptional stutter, the 7 or 8 A's, are not modified, and consequently, what ends up happening is you end up with a truncated form of that glycoprotein, a soluble form that's secreted into the body. And that's a really interesting phenomenon. So you have this kind of defective form. It's speculated it might be kind of like a smoke bomb or an immune decoy. Releases it out so the host immune response wastes its antibodies on the soluble form while it replicates. Regardless, this is something that we know exists within filoviruses and allows us to really target it. So in collaboration with US AMRID, the US Army Infectious Disease Research Center, they have BSL-4 facilities. We undertook a study where we infected rhesus macaques, non-human primates, with Ebola virus. And then we monitored RT-PCR for the virus, as well as that marker, that soluble glycoprotein that's getting released very early and very quickly. We'll highlight on just one of these rhesus macaques to kind of emphasize what we're seeing here. But on the orange y-axis, that's the level of a soluble glycoprotein using ELISA we built in the lab. On the right are the genome copies of the virus itself. So RT-PCR standard of care. You'll notice a couple of interesting trends. The first is that our soluble glycoprotein is increasing even before RT-PCR is positive. The other trend is that high levels of the soluble glycoprotein, and typically, when you go above 1,000 nanograms per mil, are indicative of poor prognosis. Doesn't matter what you treat them with. If your levels hit 1,000 nanograms per mil, you can be certain that you will pass away. So what's interesting to us is not only do we have a potential prognostic marker, hey, are you going to do well or poorly, but you also have a very early and potentially earlier diagnostic marker for infection. That got us really excited, and we decided to leverage a technology to take advantage of that unique biology to enable a really great clinical assay. And we utilize in our group a technique or sensors known as whispering gallery mode devices. Whispering gallery modes are named after this phenomena you see in architecture. There's one outside of McGall Hall here on campus where you can actually stand on a grate and kind of hear these echoes. This is an image of St. Paul's Cathedral in London. Inside this circular gallery, you can stand on one side and whisper to someone on the other side, and they'll hear you very clearly. And the idea behind this is that the sound waves are getting trapped within that circular structure, analogous to kind of total internal reflection. We're going to take advantage of the same technique, this idea of resonance, though. This is mechanical resonance. You're adding two waves in phase with each other. On the right is the Tacoma Narrows Bridge up in Seattle, an example of mechanical resonance gone catastrophically. So we're going to take that same idea, but leverage it using optics. A typical setup for an experiment is shown here. We have a tunable laser source, a fiber taper that's going to enable us to couple light in and out of a resonator device via an evanescent field coupling, a resonator itself, which is typically on the micron scale, and then a photodetector. In this schematic, this is our fiber taper and a resonator device. If we meet this resonance condition, m lambda equals 2 pi r effective, so m is just going to be a non-zero integer, lambda is going to be the wavelength of light we're coupling in, r is the radius of that structure, and then ineffective, and that's really the critical component, your effective refractive index. If you meet that condition, you have these characteristic resonance peaks, and if you scan across a number of wavelengths, you'll see these characteristic Lorentzian dips. In contrast, if you don't meet that resonance condition, no light is coupled into that resonance structure, you end up with essentially a noise background. How do we use this for sensing purposes? We're essentially going to now coat these devices with a capture agent of any kind, so an antibody, an aptamer, a target across many different things. Antigen of interest binds, changes that local refractive index, and that's evidenced as a shift in that resonance peak, which we can track in real time, very analogous to like Biacore measurements or SPRI. I will say there are a number of different geometries we can use for these. Today we're going to focus on rings and bubbles, but just keep in mind these come in a number of different flavors depending on which application you want to use. It's, yeah, right at the interface. Typically for sensing purposes, it'll be right at the surface within the evanescent field, typically on the scale of hundreds of nanometers. This is it's not a really practical application, but this is a 3-photon up conversion, so we can visualize an actual buildup of resonance in one of these cavities. Highlighted there in red is the actual fiber taper that we're coupling light into a toroid device. But you can see, once again, this buildup of power inside that cavity. For our first set of experiments, we stuck with chip-based devices. These were co-developed by a company, Genelite, that we helped found and actually develop a lot of that tech. This is a chip-based system. So on each individual chip, that's 4 by 6 millimeters, we've got 128 individually addressable sensors. We're repurposing a lot of tech. We're working in the C-band in essentially 1550 nanometers, cheaper tunable lasers, and honestly, we can outsource a lot of the fabrication to commercial foundries to generate these chips. For fluidic handling, we're using a cast molded plastic to really control fluid on top of the devices themselves. And you know, this is something we include a lot when we give the talk. So pretty much we're confining the light, we're getting signal amplification, and we're getting our sensitivity really from those multiple interactions that we're seeing. We can, of course, coat our surfaces with a number of different chemistries. We work with silane-based chemistries pretty extensively. We just coat the devices with silanes, use primary amines with the antibodies to directly conjugate. This photo is a little misleading in that the antibodies are kind of randomly assorted onto the surface, so we don't have a great orientation, but we do have a pretty robust chemistry to actually covalently attach capture agents to the surface there. For this assay, this is a essentially a modified sandwich assay. We start off with a bare surface with rings functionalized with antibodies for that soluble glycoprotein and other targets. We then add the solution of interest containing that soluble glycoprotein. We add a secondary panfiloviral antibody that's been biotinylated. And we added an additional step with these sensors with a horseradish peroxidase. And what this does is it crashes out a precipitate onto the surface. They call it a snowflake method, but essentially we're just crashing down precipitate to increase mass on the surface. How this looks like in practice is actually highlighted here, which is relative shift on the y-axis, time on the x-axis. And what we're showing there in the bold lines is actually the average of 36 individual rings. That spread is the spread we're seeing of signal for 36 separate rings for a positive and negative signal. When we're building a diagnostic assay, we, of course, care about specificity. And by specificity, we mean for the antibodies and targets of interest. So we coated our sensors with antibodies against EBOV, which is the Zaire Ebola virus, and then SUDV is the Sudan variant. It's the closest variant we have that has a soluble glycoprotein. And then vertically, we're adding to the same chip the Zaire sense, the EBOV soluble glycoprotein. We're seeing those sensors respond, the other ones responding similarly to our negative controls. We can reverse that experiment, switch our target, and see that we're getting pretty high specificity with the Sudan virus. We, of course, care about quantitating, so we can generate calibration curves off of these. Each of those data points is 72 individual technical replicates for each concentration for both the Zaire sense, the Sudan sense, and then also a control there. So we can quantitate, we can generate data off of that. We generate a limit of detection in 1% sera of low nanogram per mil, which is still about 3 orders of magnitude better than a lot of our conventional assays right now. We can increase the amount of sera as well that we're putting these into, and our limit of detection doesn't decline significantly. So there's a long explanation on why we actually don't see a massive decline dealing with the blocking, but we actually see pretty good performance parameters when we go up to higher amounts of sera as well. The real question, though, is how does this actually compare? How do we actually compare to the gold standard? And this is something every clinician is going to ask, which is, well, how are you better? So what we did is those same rhesus macaques, the same non-human primates you saw before, we took samples, ran RT-PCR on them, and then ran them on our assay paired. And what you'll see on the left is the soluble glycoprotein for all of our samples. You can see a pretty significant spread in the concentration. And then we can split it out on those that were PCR negative and PCR positive. PCR, the gold standard, was able to identify 22 out of the 30 positive infection rates. We were able to get 29 out of the 30. So pretty significant clinical sensitivity that we're improving on that alone. So very, very excited on that front. And part of that's due to the biology of the marker. Part of it's due to the fact that we've got a much more sensitive technique that we can now leverage for this. To kind of wrap up this specific section, so we have a unique diagnostic marker for Ebola virus. We have a photonic platform that's sensitive and specific. In theory, can detect earlier than RT-PCR, and I give that caveat because we honestly need additional studies. It is a difficult sample to obtain, given the nature of it. And really what we're trying to do is now move this to smaller systems. So we've been working on miniaturizing our platforms really just to portable and handheld devices that enable us to do what we're doing with the tabletop platform down into something much smaller. We've also in the lab have started working in a new class of resonant devices known as microbubble resonators. So we talked about kind of these chip-based devices, and the quality factor on those is typically 10 to the 4. Microbubble resonators have the advantage that you can have a quality factor as high as 10 to the 8th. We did switch our tunable wavelength to 780 to work compatible with water. But really why we wanted to do this is you have integration of your optical and fluidic components into a single device. So that is exceptionally difficult to do with high Q devices in general. One of the first things we wanted to do is just fabricate these in-house. We ended up coming up with our own method to actually fabricate these devices using 3D printed stages and off-the-shelf components. So essentially what we do is we take a CO2 laser, we've got a rotating motor with an actual glass capillary, we've added some randomization functions into that. And what we can do is we can control both the pressure as well as the voltage to generate these very symmetric bubbles at a millimeter scale. And I apologize, those scale bars are 500 microns on that diagram to the right. So we can pretty reproducibly start generating these devices with a, pretty much, I call it a George Jetson approach. We set a capillary in, we press a button, we come back about 30 minutes later, and we've got a working device at this point. This is just a few more details on that. We can skip over some of these in the interest of time. What's great about these larger devices is we talked about, quality factor, Q factor, and it turns out as you make your devices larger, that actual shallower surface helps actually reduce some of your scattering losses. And in turn, you do end up with actually higher Q factors. We also can control our optical spectral range. So our free spectral range, our modal density is affected by the size of these, as you'd expect. And as we increase the bubbles themselves, we see the modal density changing as well as the mode excitation changing for each of these devices. These are the same exact component. We're just expanding and then remeasuring the Q factor on each of these. And you can see we're maintaining relatively high Q factors. We're still not at 10 to the 8th at this time, but we're breaking 10 to the 7, which is getting us really excited. Another component we are working on is really these micro droplet resonators. So rather than using now a glass surface, we're now using actual droplets of liquid as the resonant device itself. So actually using the droplets, which have incredibly smooth surfaces due to the surface tension, and using that as a way to really trap light. And I just showed this one slide. We've got a little bit more we'll cover on that as well. Going back, we have done some really exciting work in terms of mode distribution topography. Turns out since we're rotating these and looking at them under a microscope, we can actually determine the topography itself and reconstruct what the topography should actually be. And now we're inputting this into COMSOL to really start determining the optical modeling of these, just using our methods for machine learning and tomography to determine what that surface looks like. So sorry, I know there is a lot of information here. It's kind of a grab bag, but it is really exciting once you get to these higher sensitivity paradigms. One of the last things I wanted to mention is, why do I keep bringing up Q factor? And one of the things we're really interested in is this phenomenon known as mode splitting. So we talked about monitoring a resonant shift of these modes as our bulk solution or bulk environment changes. It turns out that you can do nanoparticle detection and sizing label-free by using what's called mode splitting. So the addition of a small nanoparticle shown down here at the bottom left, this was some work done by one of my postdoc advisors, Lan Yang. If you introduce a small nanoparticle to the surface of one of these resonators, it acts as a mirror. And you end up actually splitting your signal into a clockwise and counterclockwise component. So instead of having a single resonance mode, you're splitting it into two degenerate modes, essentially one going clockwise, one counterclockwise with each other. And instead of having kind of this bulk sensing that we see with, say, antigen detection or refractive index change, you can now do stochastic sensing of individual particles. What's exciting about this, and we've decided to integrate this, I apologize, this is a lot of data and we'll condense it in the next slide, is we're showing that we can now use these microbubble resonators to do direct detection of nanoparticles and in turn virus particles. So taking this image, these are actually spectra from air, DI water, and then increasing concentrations of nanoparticles. What we're actually seeing on the figure to the left as we go from that black figure left is the evolution of these split modes in real time between, we often use symmetric and asymmetric, but we're actually seeing evolution of these modes as we add more particles, which tell us that we're seeing an increase in the splitting. So for us, once again, very, very exciting direction that we're going into really with these devices as well. I'd mentioned the microdroplets as well with these integrated fluidics. We can control the sizes of these fairly well. This is just a silica capillary. We ended up using silanes to create a hydrophobic surface. Turns out our best silane was this commercial Rain X product you can get from AutoZone for $5. Commercial grade silane that actually enables us really, really well to control sizing. And this is another direction we're going into. So using these kind of droplet cavities with high Q to start enabling us to really look at different sensing paradigms at this point. So that's kind of our grab bag and what we've been focusing on the WGM front Before I move on, any questions, concerns, thoughts? Yes.
Hi, Q factor. So, but you say only 10 to the power 4. Is that?
Correct. So there's two catches to that. So, and just for anyone who may not know, Q factor is a quality factor, a measure of, kind of optical finesse and essentially how long does light stay in your cavity. And that's what fundamentally allows us to do a lot of our sensing mechanisms. The chips that we ended up using with Genelite were a little tricky in the sense that we are limited to 10 to the 4, one, because we're on the chip. So you're losing degrees of freedom with that movement of light. But second is we are not using silicon nitride. You can get high Q factors with silicon nitride surfaces. Those tend to actually fabricate very smoothly and have very limited scattering losses, but they're incompatible with water. The other catch with those is because we're working at 1550 with these, and I apologize, we're jumping back a lot, the absorption of water bands at that wavelength really just leads to some significant mode broadening that we're seeing. I would love to go like silicon nitride. The problem is as soon as you put water onto that, you end up creating sulfuric acid essentially.
And another question is actually, I want to ask you about comparison between the plasmonic sensor, but it seems like you're talking about the next topic or something like that.
Oh yeah, so we have some plasmonic sensors as well, and the truth is, I love really the potential of a lot of the photonic resonators. From a practical application, they're a little further off, I think. Plasmonic, we're actually getting very, very high sensitivity with some of our techniques, and we're already deploying them in our clinics now. So I would say we're actually getting better sensitivity with plasmonics at this point. Yes.
About when you start making commercial device, you obviously have to scale this, right? So being the sensitivity radius is really important, obviously. How do you deal with variation in manufacturing, especially with water droplets and things? Maybe they evaporate at the time. All of that.
Absolutely. So with the chip-based, those are a little easier, with the commercial foundry partners. And one thing I didn't mention is we incorporate a lot of our variation tends to be much more significant on the biological side with their antibodies and sensing than it is with the fabrication. For the droplets and for the bubbles, that is a huge issue. And a lot of that work with the rotating stage was an example of us trying to standardize it. I don't have a good solution. I think it's a huge, huge issue. And for us, those are still very, I would say, very naive fields of research that we're trying to go into. We're still struggling to get consistent sizes with our bubbles as well as with our droplets. If you have suggestions, we're absolutely open. There was a group out of University of Utah that was doing cast molding with pillars to get their droplets at consistent sizes. And we've got a collaborator at Technion we're working with who uses a plasma technique, electrical plasma, to create nano bridges of water that tend to self-sustain. But I don't have a good solution, to be honest. No, but great question. So moving on to kind of a different direction, we talked about how we utilized photonic resonators as a way to improve our diagnostics. We also are focused a lot on leveraging plasmonics. Before you retired, I worked with Rob Korn as an undergrad, never got away from kind of the plasmonic itch here. And we're going to take a similar approach. So what's a critical gap in diagnostics that we can leverage using different optical techniques to really bring into the clinical assays? And we're going to switch from kind of going to direct detection to serological assays. And serology is you've been infected by something, you have an immune response, and we're going to profile that immune response. Why do we care? It lets you do spatial recognition of where the diseases are, who's been infected, who's had vaccination responses. It's another way for us to really allocate healthcare resources. But in addition to that, it's also BSL-2 compatible. So you can actually take everything in a serological assay, even with the HIV and Hep B, treat it, keep it in BSL-2, and rerun it. So there are a lot of advantages. Like many groups, we did have to pivot to SARS-CoV-2 during the pandemic to stay afloat and productive. And I mentioned this is the SARS-CoV-2 assay. We retired it recently from the hospital in the last few months. And it's detecting your response against the N protein from SARS-CoV-2. Your sensitivity is between 0 and 94% if you're infected. Terrible assay. You're better off flipping a coin in most cases. And it depended on your immune status. It depended on if you had been infected. It depended on when you sampled. And this is really our commercial like kind of status. Some of our collaborators of Hong Kong University, Malik Paris and Sophie Valkenburg, showed that, hey, instead of looking at a single protein, let's start looking at a repertoire of proteins in that serological response. And you can boost your sensitivity and specificity by just looking at different biological targets. And one of my colleagues, Chao Wu, took this one step further and said, hey, we're at a point where we can engineer protein fragments. We can now look at specific responses of those fragments instead of looking at intact proteins. And we actually see, a drastic difference in sensitivity and specificity depending on which fragment you're looking at of that protein. And you can actually see an evolution of response over time of these versus, say, someone who's been recently infected versus long-term. So if you can now engineer different protein fragments, you can generate better serology. We decided to take advantage of this in some of our plasmonic tech. And we work with plasmonic floors. We've paired up with Srikanth Singamamini's company out of WashU, and they generate these plasmonically enhanced fluorophores. We start with a gold nanorod. We add a silver coating outside of that. We then add a polymer spacer that allows us to control a lot of the Stokes shifts there. And then we can coat it with pretty much any biomolecular moiety we need to. The advantages from a clinical perspective, it's brighter fluorescence. We're talking typically 10,000-fold increase, which means higher analytical sensitivity, lower sample volumes. I mentioned remnant samples. So as a practicing pathologist, when you are tested at the hospital, there if you're more sensitive, you can kind of play with all of these factors and build better assays. So one of the first experiments we wanted to do was we hand spotted 3 separate antigens, S1, the RNA binding domain, S2 and N, as well as a control protein. You can tell they're hand spotted, and it really did depend on if you drink coffee that day or not. But you can see the IgM, IgG, and IgA immune responses from patients in each of those wells. And as a clinician, we want to know positive and negative. Can you use cutoff points? You can do that with this. But we decided to take advantage of the fact that we've got piezoelectric microarray spotters that we can leverage. This is actually an image of one of the 7x7 arrays of antigens that we can print inside a single well here. And take advantage of the fact that we are now at a point where we can engineer protein domains. And instead of just looking at the N protein, let's start mix and matching different components and different targets now. This is a very typical workflow for kind of serology. You start off with your plate, in this case, a 96-well plate. Inside a single well, we have a 7x7 array of antigens. We use 1 microliter of patient sample, diluted up to 100 microliter. So the ask is very minimal. We add secondary antibodies that are against the human antibodies, and then we can add our fluorescent probes. And what you're seeing here is actually, once again, IgG, IgA, IgM for a massive amount of patients. These are 3 separate plates. We can generate what are called receiver operator characteristic curves. So your sensitivity and specificity, really for that assay clinically. So allows you to determine a cutoff relatively simply. May not be the most practicable, but it's a quick and easy way. And then we can generate, of course, serological response for each of these combinations. So instead of looking at 1 marker in a single well, we're now looking at 24 separate markers for infection at this point. We can, of course, look at different combinations. And if we focus on these responses, this is the response to the full-length N protein. We can do IgM, IgG, IgA. And if we focus on just one of these are the serological responses for someone who was hospitalized with COVID. I apologize, it's cut off on the bottom, but that is days since symptom onset on the x-axis. On the y-axis is intensity with our probes. And you can see a couple of interesting trends. Number one, there are people on day 0 who are positive by serology. Means they were either infected previously, there's some cross-reactivity, or we are picking up a very early infection that really was becoming symptomatic. Number 2, you're seeing a pretty drastic number of these points that are coming positive. So as compared to the assay that's 0 to 94%, we're seeing a massive boost in sensitivity here that we can leverage. We can do this for every combination, but what's more interesting to me is because we now have access to hospital data and time course data and remnant samples. So once again, we can pull samples straight off the lines through our broad IRBs that we have at the hospital. What we can do is actually look at trends over time for patients who are hospitalized. The top row and bottom row are two separate patients, the IgM, IgG, and IgA response. So what we're showing in each of those is their serological response to infection as a function of time to each of those markers. There's a lot to take in here, but really what I want you to notice is that the curves just don't look the same. Every patient responds differently serologically to every response. And once again, in terms of workflow, this is very minimal for us to incorporate, and we're getting useful information now. So the patient on top for IgA has a much less robust response than, say, the patient on the bottom for IgA. So you can start looking at a different way of approaching medicine and really trending these characteristics and trends. I did mention, kind of the goal is for detection of pathogen X. We showed 2 virus applications as well. One of the projects that we're ongoing right now at the hospital is the multiplex detection of cytokines. Cytokines are pretty much what your immune system uses to modulate responses and compute. These are transient. The concentrations can often be incredibly low, make these diagnostics exceptionally difficult. And so now what we've done is take advantage of the same techniques, plug and play. We are spotting antibodies at this point. And then what we're doing is actually now looking at cytokine concentrations. We have 12 separate cytokines we've actually arrayed. That's an intensity map of IF and gamma. But in each of these individual wells, we're targeting 12 separate cytokines. And once again, 1 microliter of sample that we're essentially just building out into this assay. This is some of our preliminary data that we're really pushing. These are patients who are septic. So we can identify in our system they have a massive bacterial infection. And now we can start building up models to say, look, who has sepsis, who doesn't have sepsis based on these markers. And for that, this is incredibly exciting. So sepsis is still a clinical diagnosis right now. It's based on symptoms. You can see the patients in red up top are the ones who are septic, actively infected. The ones in blue are quote unquote normal patients. So we can start changing the way we're diagnosing because we've got access to these highly, highly sensitive techniques. So hopefully, I've highlighted that, look, we can do quite a bit with the serological testing and our multiplexing capabilities. The last thing I really want to highlight is that we're still moving towards miniaturization with these as well. And one of our big pushes is assay development in resource-limited settings. This was me a few years ago in Cape Coast, Ghana, in one of the slums that we work in. And when I mean resource-limited, to the right is one of our clinics. That is one of our clinics. There's no electricity. There are no gloves. You're lucky if you have a technical person at all, but usually it's just a local who is there to help. So what we mean by resource limited is no electricity, no refrigeration, no technical expertise whatsoever, and no equipment that's going to help you run these. And what we've been moving towards and what we think is a good compromise are lateral flow assays. So combining these plasmonic, fluor enhanced, probes as well as lateral flow. So using that same Ebola test that we saw before, we're going to use the same markers, but just convert this now to a lateral flow assay format. In this image, we're mixing the sample with a solution containing the plasmonic fluor antibodies that buys us a little bit of sensitivity and cleanliness. We then put it onto one of our flow pads, which is a sample pad. Capillary action whisks it up. We've got 2 target strips, a test strip, and then our absorption pad at the end. We can generate calibration curves for those same 2 Ebola viruses, the Zaire sense and the Sudan sense. We can pre-incubate for longer periods of time to increase our sensitivity. And if you have a total assay time of 30 minutes, we're getting sub-nanogram per mil detection, actually beating our photonic resonator platform quite a bit. So really, really exciting on that front. As expected, you do see a little bit of hook effect, kind of that calibration curve begins to decrease as your concentrations go up. But once again, very, very excited about this. And our clinical sensitivity, using those same samples you saw before of those infected primates, we're now at 100% clinical sensitivity with this assay. We can do direct detection from serum as well. So this is if we, instead of pre-incubating, we just add everything into the sample immediately. We actually print the floors onto the strips themselves, and we're still getting low nanogram per mil sensitivity. So sufficient for all clinical applications on our end. And we've been working with Brightest Bio, an early stage startup who we've been submitting some SBIR, STTRs with, to really build a portable system. And this is one of the prototypes that we have going. And the idea is a shoebox size that we just take out with us to the clinic, and we read out with your phone. So that's really where we're moving to with a lot of these technologies. I know it's been a lot today, but hopefully the broad theme is really when we've got access to really unique optical techniques, when we've got access to lab medicine and a hospital system that enables us to utilize samples and unique biomaterials, we can really leverage this to address a lot of critical gaps in our diagnostic space. A lot of acknowledgements, a lot of people who have really helped push this work, especially on the diagnostic front, the Amarasinga Group, Leung Group, Bailey, Lan Young out of WashU, who's still incredibly supportive. A lot of our industry and Army-sponsored studies. Of course, this would not have been possible without the current group as well, who have been just amazingly patient and phenomenal scientists with me. With that, I know it's been a whirlwind, but thank you so much for coming out today and really for your attention. I'd be happy to take any questions you might have.
Thanks so much, Dave. That was fascinating. Any questions for Abe? Yeah, I think people are finding her.
Regarding the glycoprotein detection, you amplify the signal with resonance. Remind me, surface enhanced Raman spectroscopy. Any comments regarding if the surface enhanced Raman spectroscopy could be used for this detection?
It absolutely could, and there's a lot of people using the WGMs as essentially sources for SIRS. So they'll build a small resonance structure or pull it into close proximity of, say, a gold or silver kind of composite as an amplifier for a SIRS signal. So there is a possibility there. SIRS, and I know Regina Regan and Alan Hochbaum have done a lot of work on this and have had some really exciting, I work with them on the micro front. SIRS is really great, but the problem is specificity, fundamentally. So you're still going off of the Raman spectrum. So if they can address the specificity issue, I think SIRS could really push the limit in some exciting ways. It's how do you incorporate those antibodies into the SIRS? I'm not as familiar with.
Thank you.
Daryl.
Sorry, I was really interested by you said that the lateral flow assay was beating the microresonators.
Yes.
Long term, is this are you saying then basically that actually if you just include some good ideas about how to concentrate your signal and all of this, you can actually do just as well, even though your sensitivity of your individual sensor might be better for the microresonators, but actually, practically maybe that'll flow. That's the answer.
No, you've hit the elephant in the room and the nail on the head, which is there is building a sensitive device which can, and the resonators, there's people who can do single virus detection, single particle detection, and that's great. But from a practical perspective, if you tried to lug any optical setup into that, resource limited clinic in Ghana, it's never going to work. There may be paradigm shifts. The miniaturization of tunable lasers has really taken off. There may be shifts that enable us to actually get them more portable, but at this time, I don't think it's a practical solution for field deployment. I think there are still applications. One of our projects is we're doing blood coagulation using the resonators. That's a place where you need a central lab. There's a clear need. But from a practical point of view, I don't think the resonators are going to necessarily take over the clinical side on that. I think the floors and the lateral flows are just really, really simple and really robust.
I have a super naive question about I guess just thinking about the last thing you're showing in Ghana with the, where you can't have any specialized technology or anything. So one thing that your lateral flow assays needs is serum, but in terms of just isolating the serum from the blood.
So actually on the last curve, I should have mentioned it is serum. We have done whole blood detection, which is And what you can actually use on the lateral flows is they actually have separation strips that you can incorporate on. So it's essentially just a small nitrocellulose filter that enables you to remove most of the red blood cells. So Abley and I have talked a bit about this, which is there are ways we can incorporate the whole blood. The serum issue is a problem. A lot of people, there's videos on YouTube of them doing like, the helicopter way. They take a string, they're spinning it around. They try to come up with clever ways, but it is an issue. The gold would be finger prick, whole blood, 100%. I completely agree.
Yeah, I remember, I forget how long ago, but Rebecca Richards Quarterman, I remember they had, I think it was an undergraduate project, but it was basically they had used a salad, like basically just a salad spinner.
Yeah. Oh gosh,
The
yeah.
centrifuge. So I don't know if that's good. That's enough to separate blood from red blood cells out, but
I would love to see actually salad spinner would be amazing to implement. I know like some of our colleagues who would go to Ghana, they were doing water purification and they were using just, sand and soil. But like the simplest are often, the easiest implementable.
Thank you for the nice thought. So for the multi-passing pass, I'm not completely sure I understand clearly. For the 7x7 array,
Oh yeah.
are they plasmonic structure and what are the different on those
No.
arrays?
So for the 7x7 arrays, we're either spotting antigens or antibodies onto there. The plasmonic component is only the readout with a fluorescence. And so what we're doing is spotting either antigens for the serological response, or we're spotting antibodies for our cytokine assay. The advantage of the floors is that we can then use a secondary antibody and one readout signal. And our multiplexing comes from the spatial distribution of those sites. Surprisingly, we get better signal to noise with an array than with an entire well. And we think a lot of it has to do with just kind of that confinement of the signal. We've noticed just tends to give us better signal to noise than just a single well with a single target. So, great question.
I have another super naive question about like specifically about the microresonators. Can that be done in an, I don't know what it would, could that sort of configuration or sensing be done in an imaging configuration? So if you put like let's say a really thin tissue sample on.
Absolutely. And I know, so Lan Young actually founded a company, DeepSight, that I don't know if they're still using these. They're using them as imaging receivers. So, we talked about binding antibodies for specificity today. The truth is these are highly sensitive to any environmental stressors. And so in theory, they're sensitive to, say, vibrations or scattering or any type of mechanical signal you could have. So people have done embedded tips on fibers. They've also used these. I know Bowen out of University of Queensland has done a lot where you use these as receivers for like ultrasound or OCT. And so you can use them for other ways. They are pretty versatile, and it's a direction we'd absolutely love to explore.
Okay, I think with that, let's thank you.
Thank you so much.
Wonderful talk.